This is now a continuation of the immune system after the first one, the innate and body defenses. We're now going to get into the adaptive body defenses. The adaptive immune system is very specific. It's a specific defense system that eliminates almost any pathogen or abnormal cell in the body. One of its activities only that is related to the previous discussion that we had on the innate is that it amplifies inflammatory response and it activates the complement system. Both of these two are part of the innate, but they are being amplified and being stimulated by the adaptive defenses. But there is a shortcoming for the adaptive defense in that they need some time, that they need to be primed by initial exposure. They need to know exactly who or what they're actually dealing with, and it takes time for them to know it. So the priming itself takes time. Now, the adaptive immunity, though, is very specific. It will recognize and it will target very specifically the antigen, and it is systemic. It happens inside the body. It's not just outside, just like acting as a wall. No, it is actually inside. It is not just restricted to the initial site, which is outside the body. Once they get inside, then you have lots of cells that can actually defend us there specifically. And it has memory. After they mounted what we call this an attack, and if they know already how to fight the antigen, they will gain a memory of how they did it, and therefore the subsequent exposure, or a second or a third or subsequent exposures, will already be something that you could guarantee defense with. And there are two main branches of the adaptive system. One is what you call the antibody mediated. This is actually the production of antibody that is being secreted in the blood. That's why you call it also humoral immunity. And the other one is cell mediated immunity, which is actually the cellular part. The humoral immunity, this will be your lymphocytes forming the antibodies. They are freely circulating the antibodies in the blood. They bind temporarily to the target cell, and they would temporarily inactivate the action of the target cell. They are also marking the target cell for destruction by phagocytes or by the complement system. So the humoral immunity has extracellular targets. So these are just like bullets. Okay, they are formed by specific cells, and you call them the B cells, and they are bound to pretty much target a cell that is holding an antigen, a foreign cell. So there's the second aspect of the adaptive defense, which is our cellular immunity. Here, the lymphocytes act against the target cell. There is actually one-on-one combat. They are actually killing directly the infected cells. Okay, there's also indirect killing. It's not really that they have hands that they could actually fight. No, they are actually doing this direct killing indirectly by releasing chemicals that would enhance inflammatory response or activating other lymphocytes or macrophages so they can actually do the job. The cellular immunity pretty much has cellular targets. So I have been talking about antigens. Well, what exactly are antigens? Antigens are substances that can mobilize adaptive defense and provoke an immune response. And if they provoke an immune response, does that mean that antigens are actually foreign? Well, put it this way, guys. We have our own antigens, and self-antigens to us, they're not foreign because they are ourself. But when our antigens are actually transferred to another person's body, then they become foreign antigens. So every single one of us has foreign antigens or has antigens, and is supposedly recognized by our white blood cells to be part of us. and therefore they are self. But there are also other self antigens that are self to others, but when they get transferred to us, they become foreign. Okay, so they could be when they are transferred to us, and they provoke an immune response. When you say provoke an immune response, that means our own white blood cell will react to it, being that they recognize that it is foreign. They have to fight it so that it's not going to be part of us, because they know exactly what is part of us. So anything that can mobilize this particular reaction of our defense mechanism is called an antigen. Antigens are targets of the adaptive immune response. So whenever a specific immune response cell, a B cell, will defend us, it is always looking for the antigen. They will have to know what the antigen is. So any bearer of the foreign antigen is actually going to be a foreign cell that they know they'll have to kill. So most of the antigens are large and they're complex molecules, and they're not normally found in the body. And pretty much characters of antigens, it can be a complete antigen, that means they are bound to a big protein. Therefore, they're very visible. Or they are incomplete, and when they're bound to very small proteins, you call them haptens, and they contain antigenic terminants, and they are not always like foreign. They could be your self antigen. So complete antigens have two important functional properties. They are either immunogenic, which means they have the ability to stimulate the proliferation of specific lymphocytes, and they are also reactive, which means they have the ability to react with activated lymphocytes and antibodies released by immunogenic reactions. Examples of this will be your foreign proteins, polysaccharides. These are carbohydrates, lipids, nucleic acids, seen on many foreign invaders such as pollen and microorganisms. So they do have this reactivity. So whenever you get to be like reactive and you become hypersensitive to something, that means you're reacting to it. Your white blood cells are reacting to it. Therefore, you're dealing with a complete antigen. Okay? When you stimulate further the proliferation or the number of those lymphocytes that are dealing, then you have immunogenicity. That particular antigen has immunogenicity. Therefore, they are complete antigens. The incomplete antigens are the smaller ones. You call them haptens. The involved molecule here is clinging to a very small protein, and they don't seem to be immunogenic just by themselves. So the protein here are small peptides, nucleotides, and some hormones. But at some point in time, these haptens can actually, by way of evolving, can attach itself to our own body protein that is big. And a combination of this protein and hapten will be making that particular antigen bigger, and then they become visible. And then they can actually, now they can be, immunogenic, and they can also be reactive. Example of this is when at the beginning of, let's say, 5 years, so when I came to the United States, I did not have any allergic reaction to the pollen here. But after 5 years, and it's because the pollen here, to me, when my body takes it, is that they're really near haptens. But after 5 years of taking it all the time, every year, then they were able to pick up my big proteins, and then after that, my body recognized it already, not anymore as a hapten. They become immunogenic. Then my body is interacting with it now. So they become very reactive, and my white blood cells are reacting to it. And that's when I started having hay fever after 5 years of being here in the United States. But when I first came in here, I didn't have it. I just developed it, and that's how you develop allergy after several years. So they cause the immune system to mount an attack that is harmful to the person because it attacks self-proteins. Okay? An example of this will be your poison ivy or animal dander, detergents, cosmetics. Okay? So one of the character of complete antigens will be the presence of what they call antigenic determinants. What are antigenic determinants? Well, antigenic determinants is an appendage. It's a part of the antigen itself that the antibody or lymphocyte receptor would have to bind into. And usually, the naturally occurring antigens have many antigenic determinants. The more antigenic determinants an antigen would have, the better it is for our body to get rid of it, to neutralize it. However, if it has only one antigenic determinant, then it's going to be a problem because you will need the correct white blood cell to look for just one correct white blood cell that would genetically carry the DNA that would neutralize that one antigenic determinant. So it is harder if it has only one uh, antigenic determinant. It'll be, if it has several antigenic determinant, then there could be several white blood cells. This is actually like an antigen with several, three different types of antigenic determinant. And these antigenic determinants are like shaped like in different ways. So here's one antibody. This is another antibody. This is another antibody. So here, even if you're talking only of, let's say, two antibodies that were produced by two separate white blood cells, then you can already like pretty much like disable this antigen here. But if you have only one, and it takes a while to find that lone white blood cell that can produce the specific um, antigen, well, antibody to fight this, it's going to be very hard for our body to look for that. So it is better if the microorganism or the antigen bearer will have, or the antigen itself will have several antigenic determinants. And usually in nature, they do have several antigenic determinants. Now let's take a look at our own antigen. Our own antigen we call self antigen. They are covered also with a variety of proteins that are located on the surface of our cells. But they are not antigenic to us. To us, they are just regular fixtures around our cells that our white blood cell know already, and therefore they're so used to it because it's their home, all right? But to others, they may be antigenic to others. For instance, in terms of whatever you are actually giving transfusions, as part of you is actually your blood is being offered as a transfusion, or your own body cells or tissues are actually offered as what you call this, a graft. Okay, so there's one important set of cell proteins, okay, which are actually in the group of glycoproteins. You call them major histocompatibility complex. This is your MHC proteins. They are coded by the MHC genes and is actually unique to individuals. So our genes, you and me and all of our classmates, would have different MHC makeup because we have different DNAs for our MHCs. These MHCs contain groups that can hold piece of self-antigen or foreign antigen. The T lymphocytes can recognize only antigens that are presented on the MHC. Remember that, guys, because this will probably come up in your exam. So the adaptive immune system will involve three different types of cells. You have the two lymphocytes, the B cell, which is the one that produces the antibody, the T cells, which will be the one that is involved in cellular immunity, and the antigen presenting cells. Usually the antigen presenting cells do not respond to specific antigen. They play a very crucial, although your book would mention it as auxiliary role, but very crucial to me. These are my heroes. The antigen, most of the time, the antigen presenting cells, if they will not be doing their work properly, the B and the T lymphocytes are going to be lost. They probably will have minimized defense mechanism. They actually have a very important role in the defense. So, lymphocytes. Well, guys, just want to make sure that you know the lymphocytes, the white blood cells. It's just a review. They originate in the bone marrow. Your great-great-grandmother of all blood cells are your hemocytoblasts, and your lymphocytes in the bone marrow originates from the same great stem cell. All right, and from there, from there, after differentiating and being committed to become lymphocyte, they are now going to branch into two. They will branch into two. These are lymphocyte precursors. They will branch into, my bad. They will branch into two. One will go and proceed to the thymus gland, and there it will be um, uh, gaining immunocompetency in the thymus gland. This group will be your T cells. The other group will go and will actually just stay in the bone marrow, and these will be your B cells. These are the ones committed to become B cells. And both of them, once they are released in the bloodstream and they are already immunocompetent, will now be staying in their hub, which is actually the lymph node. Okay? They actually travel from the hub into the bloodstream if they're needed in the bloodstream. Okay, there you go. And then they go back into the hub if they are not needed anymore. So there are other things that they do whenever they go out or they get out of the lymph node. Once they receive the information that there is an infection, they move out and they clone themselves. If they're able to encounter the microorganism and the antigenic marker, and then they are the ones that are committed, or they carry the DNA, the gene, to form, let's say, the antibody. And there's only one lymphocyte that can actually do that. You cannot kill millions of germs with one lymphocyte. So what's going to happen is that it needs to be cloning itself. Okay, so the cloning will happen after the exposure, and those millions of clones or thousands, hundreds and thousands of clones that will be formed are the ones that are going to be producing enough antibodies for our defense. Or if they are helper, if they are actually the T cells, then they become the killer T cells only after exposure to the antigen. These specific cells will not spring into action unless they know what they're fighting and they know that they can deal with the with the microorganism, with the antigen. So, in the process of maturation, these lymphocytes are educated in between 2 or 3 days, and they mature in the primary, what you call primary lymphoid organs. The B cells in the bone marrow, that's a primary lymphoid organ of the B cells, and the T cells will be in the thymus gland. And the lymphocytes are educated for two reasons. First reason is they need to gain immunocompetence. Lymphocytes must be able to recognize only one specific antigen. All right? Only one specific antigen. Therefore, you would need, if you will have 100,000 different antigens that probably our body has encountered our entire lifetime, you will need to have those 100,000 lymphocytes around because they recognize only one specific antigen. B or T cells display only one unique type of antigen receptor on the surface when they mature, so they can only bind to one specific antigen. Then your lymphocyte also has the what you call self-tolerance. Lymphocytes must be unresponsive to own antigen. That's the first thing that they learn before they are released into the bloodstream. They must know what self is. If they will not be able to recognize self, they will undergo apoptosis. They will be killed right there, either in the thymus gland or in the bone marrow. They will not be released. Why? Because they will, if they are not tolerant to self, they would most likely be killing self. That's not good. That's an autoimmune response, and you don't like that. Okay, so after staying in the primary lymphoid organ, which is your bone marrow and the thymus gland, when they are released, they're released in the bloodstream. They don't stay in the bloodstream. They have to pretty much stay in hubs. And this is actually what is meant by this, seeding the secondary lymphoid organs and circulation. So when you have already the immunocompetent B cells and T cells that has been released, but they are still naive, they have not been exposed to the antigen, they're exported from the primary lymphoid organ, which is the bone marrow and the thymus, to seed or colonize the secondary lymphoid organ, the lymph nodes and your spleen, okay? This will increase the chance of encounter with antigen. So they may not be floating in the blood because they're not necessarily in the blood yet, but they should actually stay in different hubs. They don't just stay in the lymph nodes or the bone marrow, just the thymus or whatever. They have to go out somewhere. As a matter of fact, your thymus gland doesn't stay longer than 11 years. Your thymus gland, after 11 years, when you're at age 11, your thymus gland would probably be very small or would already be disappearing. Okay? So the first 11 years, your thymus gland will be active. After that, it's gone. Okay. After seeding, your antigen will encounter. Your naive antigen will now be ready to encounter your foreign antigen and be activated. So these naive lymphocytes first encounter with antigen will trigger the lymphocyte to develop further, to become more committed. This time, they are not anymore rookie soldiers. They're going to become, well, maybe what you call the Navy SEALs, or they will become part of a really more a more progressive soldier, a SWAT team or something. So your lymphocyte is selected to differentiate into an active cell by binding into a specific antigen. The moment it binds into an antigen and would realize that, okay, this is the specific antigen I can tackle, okay, then it is already ready to clone itself. Now, this is actually referred to as a clonal selection. If the signals are correct, the lymphocyte will complete its differentiation into an active cell. But because there's only one of it, there's only one of the lymphocyte that has recognized it, that lymphocyte will need to do like a good amount of, let's say, antibody. It will need to have several of itself. So it will clone itself. That's when a lot of it will be released into the blood and will be formed as plasma cells, call it, to form the antibodies. These will be the clones itself. So the clones, this is now, the next one, proliferation and differentiation. Once they are selected, activated, the lymphocyte will now proliferate, and these proliferated lymphocytes are what you call the clones. Most clones are effector cells, and they are the ones that are actively fighting infection. But then after the battle, after the battleground has already eliminated your microorganism, then you don't need them anymore. And when you don't need them anymore, you don't want them staying in the area. So there will be a group of cells, the suppressor T cell, that would suppress them and would be producing something to eliminate them. And before that thing will happen, some of them will remain as memory cells and will hide again in their hubs, in the nooks and crannies, carry the memory of the antigen, the antigenic marker. And that enables them to respond to the same antigen more quickly the second time it is encountered. Both the B and the T cell memory cells and effector T cells actually would circulate continuously. Okay, but some of them pretty much can stay in the hubs, just like anything else. So, genes, and this is what I tell you, genes, and not the antigen, will determine which foreign substance our immune system will recognize. And guys, if we do not carry the genes that will pretty much recognize the antigen and how we can battle the antigen, we're not going to be able to defend ourselves if we do not have the genes for it. And we will just die. So a variety of immune cell receptor is a result of an acquired genetic knowledge of microbes. Our body has a vast knowledge of it since the human race has actually evolved on Earth. There are about 25,000 different genetic codes gene codes for up to 1 billion different types of lymphocyte antigenic receptors. 1 billion types of lymphocyte antigenic receptors, and that's a lot. There's a huge variety of receptors, and gene segments are shuffled around, and that can result to many activation. So it is not true that one lymphocyte is only one gene. There could be that the lymphocyte will be able to shuffle around the gene, and therefore it could actually carry on the ability to produce antibodies for maybe several genes. But usually, the worst case scenario will be one lymphocyte and one gene. So T cells mature in the thymus under negative and positive clonal pressure tests. This is pretty much how they are tested when they become immunocompetent. And there's two processes. One is what you call the positive selection process. This is where selected T cells capable of recognizing major histocompatibility protein, pretty much those cannot be destroyed by apoptosis. This will be the T cells, or rather the T cells that are able to recognize the self to be self. Those will not be touched, which means this is good because they recognize self. When they are developing immunocompetent, the thing that they must be able to do will be to recognize self, and that is by way of positive selection process. They also must be able to do a negative selection process. Okay, this is actually when T cells that bind to self antigen, pretty much displayed by self MHC, is going to be prompted to be killed. This is actually something that should not happen. Which means, guys, you should not actually you should be able to recognize self. But when you recognize a self that is displayed by a self MHC, then you are going to be killed. That is actually the negative selection. And this process is what you call the clonal deletion. This particular negative selection is clonal deletion. And this will ensure that that particular T cell is actually going to be tolerated by your lymphocyte. The lymphocyte must know self. When they are released out in the bloodstream, they should know they know self. Remember that there are 1 billion antibodies right there, and there's probably only one that it can recognize. But there's one thing that it needs to recognize, and that is self. Because it cannot kill self, it cannot be antagonistic to self, and that's the reason why when they are in the spleen and in the bone marrow, the thing that these lymphocytes will need to do will be to know who is self. Now, this is a very good instructional diagram of the positive selection. All right, and this will tell you pretty much that this antigen presenting cell coming from the thymus, and this is a developing T cell, recognizes pretty much this one here recognizes the major histocompatibility complex, but it does not latches on to the self antigen. That's a self antigen right there. That's a self antigen right there. If it does not recognize the MHC, if it does not recognize the MHC, it is going to be killed by apoptosis. If it recognizes the MHC, but does not recognize the self to be, something that is something that it needs to kill, then this one pretty much is survival. It's actually it results in the survival of the lymphocyte. This lymphocyte here is going to develop good immunocompetence because it is not latching to the self antigen, which means It knows that that is a self-antigen and it should not actually latch on it and it should not kill it. All right. However, this one here is a negative selection. Take note that it latches straight onto the self right there. Recognizing the self-antigen will result in apoptosis. This eliminates the self-reactive T cells and could cause an autoimmune disease, this one here. If your T cell would recognize your self-antigen as a foreign antigen, then that particular T cell is actually going to uh, uh, be killed before it's not going to be immunocompetent. It's not going to be released outside. Before it's released outside, it has to be eliminated. And right here, it is act just like this one here. It could pretty much like uh, it is not recognizing the self. It's not binding tightly to the self antigen. This would result in survival. It could latch into the major histocompatibility complex, like this one is latching into it, but it's not recognizing the self antigen as a foreign antigen. Therefore, this type of negative selection is actually good for its immunocompetence. So let's take a look at the antigen presenting cells. And this is what I mentioned to you as my heroes. The major histocompatibility complex. We're not anymore in the major histocompatibility complex. Let's go into a type of cell which is called the antigen presenters. The antigen presenters could be anything that will eat the antigen or would have the ability to phagocytize an antigen. It could be a phagocyte, it could be a macrophage, it could be a dendritic cell, a neutrophil, right? They engulf antigens and then present fragments of the antigens to T cells for recognition. So these will be your three different types of antigen presenters. And the dendritic cells are found in connective tissues, and this is your dendritic cells. And your macrophages is actually widely distributed. They are coming from monocytes, and you know that already. And your B lymphocytes, they pretty much do not activate naive T cells, but they present antigens to helper T cells. How does the antigen presenter do this? Well, the only thing they do is to eat, okay, run after the bacteria, digest it, and those fragments of the bacteria that are antigenic, they're not going to eliminate it. They're going to expose it in their cell membrane, and it will be visible as part of themselves. Therefore, they can stay in one corner of, let's say, the lymph node and say, hey guys, I have just eaten a bacteria and you want to see a portion of the antigen? Here it is. I have it on my skin. That's how they present it. And these particular antigenic presenters this action is very vital simply because of the nature of our blood. Our blood is actually free flowing. And as it is free flowing, the bacteria flows with it, and most of our white blood cells cannot run after those bacteria, not that swiftly. So, whatever the antigen receptors or whatever bacteria an antigen presenter can actually uh, cling into and eat and pretty much digest, they can present it to white blood cells or lymphocytes straight into any of our primary and secondary lymphoid organs and present it to them so that they will know who they're dealing with. That's actually the purpose of an antigen presenter rather. So let's take a look at the activity itself. In the first activity, you call it the humoral immune response. This would involve the B cells. When a B cell encounter a foreign antigen, it can be provoked to undergo a humoral immune response. In the same in the event that it is actually provoked, then it will produce in the end an antibody that's specific to a particular antigen that's actually been in attacking us. So How does this thing happen? Well, first of all, the B cells are activated when antigens bind to the surface, and there is actually a it triggers the what do you call a process. There's a process that triggers the receptor mediated endocytosis, okay, leading to the proliferation and differentiation of B cells into effector cells, which means if the B cells would recognize, okay, he is an enemy, okay, then there's going to be a receptor mediated endocytosis of cross antigen receptor complexes. And that in itself is already the act of recognition that will lead to, hey, oh, I know how to fight this. So, but there's only one of me. So, what am I going to do? Well, I am going to reproduce myself. I'm going to clone myself so that there will be several of me, several hundreds of me that can produce the correct antibody, because we're talking about the B cells here. So these are what's called the plasma cells, the antibody secreting effector cells, and they are able to really secrete about antibodies at the rate of about 2,000 molecules per second for about 4 to 5 days, and this is going to pretty much consume all the energy, and therefore they die. So the antibodies circulate in the blood, binding to free antigen, marking them for destruction by innate mechanism or other adaptive mechanisms. All right. So here's actually your antigen, and that's actually, this will be your lymphocytes, and the lymphocytes are now the lymphocytes now has producing antibodies, and they're capable of trapping the antigen. All right, so they know that they can do this. They pretty much they are now going to proliferate to form clones, and these clones will be producing the same antibody that they know can actually kill the antigens. So clones that do not become plasma cells can become memory cells, and this will provide immunological memory that can help us conquer our battle by mounting an immediate response for future exposures to the same antigen. All right. In the primary immune response, which means when we encounter the what do you call this, the antigen for the first time, that's why you call it primary. The cell proliferation and differentiation upon exposure to an antigen for the first time is slow. There's a lag period of between 3 to 6 days. The peak plasma of antibody is reached in 10 days. So after that, then the antibody production level will decline. So this means that this lag period, what is happening in this lag period? How come it's really long? Well, you need to find out which particular white blood cell is carrying the DNA to produce the correct antibody. And therefore, for as long as that particular white blood cell is not yet around, then there won't be any antibody. So hunting down that, and that's why we become sick, and then after that, in the first exposure, we become really very sick, okay? Because our body is really looking for the right antibody, or the right lymphocyte that can produce the antibody. However, if we have already gained the immunity, and our memory cells are just hiding in the nooks and cranny, a re-exposure to the same antigen will give a faster, more prolonged, and more effective response. This is actually what's called the secondary immune response. You have the memory cells that are already pre-sensitized and carries immunological memory, and they respond within hours. It doesn't take days, and therefore you don't get sick anymore. The antibody levels will peak in 2 to 3 days at a much higher level, and so you don't feel sick anymore at all. All right. This is actually the amount of antibodies on the first few days on the first exposure, but on the second exposure to antigen A and the first exposure to antigen B. So this one here is already a second one, the secondary immune response, which is really nice and quick. This is a primary immune response to antigen A that occurs after a delay. All right. So let's take a look at the active humoral immunity. Okay. Active humoral immunity occurs when the B cell encounters antigen and produces specific antibodies against them. And there are two types of immunity which we call active. One is we call natural acquired. You acquire it by, well, it's a formed response to actual bacterial or viral infection. Okay, uh, you have to pick up the virus or the bacteria and you get sick with it and you fight it and you get sick with it. That's exactly what natural acquired is. With the artificial, that means you form a response, okay, by getting something that is not natural, and that is maybe an attenuated form of what do you call this? Attenuated form of the antigen itself, which we call as a vaccine, okay. And so this artificial acquired humoral immunity is formed in response to vaccine, okay, that is made in the lab. They provide These vaccines provide antigenic determinants that are immunogenic and reactive, but usually they will spare us the symptoms of primary response. So passive humoral immunity occurs when ready-made antibodies are introduced into the body. B cells are not challenged by antigens, and immunological memory does not occur. The protection ends when antibody degrades. And there are two types of passive humoral immunity. One is natural acquired also, and these are antibodies delivered to us via our mama, or our via the placenta, or through our mother's breast milk. Or they can be artificial. When you get sick, when a baby gets sick or when a toddler gets sick, like when my baby got sick with the measles. She was not a baby anymore. My oldest was a 4 year old at that time, but then my youngest was only 2 years old. So I was afraid for my 2 year old when my oldest got the measles. So I was afraid for both of them, but most especially I was afraid for the baby who was only 2 years old. So I brought both of them to the doctor. The doctor said, we could just let the 4 year old get the measles. She's strong enough, but the 2 year old we're going to give an immunoglobulin shot. So she was given immunoglobulin, which is an injection of the antibody itself, gamma globulin. The protection here is immediate, but it lasts only for about 1 month. And so this will be your cheat sheet, guys. Pretty much telling you what the humoral immunity is. You have the active immunity and the passive immunity. And just make sure that you know that this is something for you to review. So let's take a look at antibodies. What are antibodies? Well, they're also called immunoglobulins. They are made by the body from the gamma globulin or the immunoglobulin portion of the protein of the blood. Pretty much these antibodies are capable of specifically binding with antigens detected by the B cells. The B cells are the ones that produce this. And they are grouped into 5 classes. Guys, I'm not going to give you pretty much like so much question on what they are and the overall structure, but I want you to understand what these antibodies are. Well, actually, your antibody pretty much is shaped like a Y, like this. There you go. That's a Y shape. And know that the stem pretty much is the area that determines the antibody class, and it's considered to be the effector region of the antibody. It really pretty much like serves a common function in all antibody, dictating the type of cells or chemicals that the antibody can bind, okay, and how the antibody class functions to eliminate the antigens. That will be your stem, that it makes up pretty much the constant regions. Then you have um, you have the variable regions. Okay, the variable region, which is one end of the Y part, is actually the end that is intended to be the antigen binding site. All right. So here is here is your these are your somewhere here will be your specific antibody binding sites. And there are five classes: the IgM, IgA, antibody, immunoglobulin M, immunoglobulin A, immunoglobulin D, immunoglobulin G, and immunoglobulin E. So just make sure that you know that IgM pretty much is a potent agglutinating agent, and it readily fixes and activates the complement. And then your IgA is actually secretory and helps prevent the entry of pathogens. All right, this is your IgM and your IgA. Okay, the IgA is found in body secretions such as saliva, sweat, intestinal juice, and milk. And your IgM is truly the first immunoglobulin class secreted by the plasma cells during a primal response. Okay, this is actually very useful in diagnostic because presence of IgM in the plasma usually would indicate a current infection by the pathogen that elicits the formation of the IgM. Then you have your IgD, immunoglobulin D, G, and E. Your immunoglobulin D functions as a B cell antigen receptor, and they are found on the B cell surface. Your G is the most abundant of all the antibody in the plasma. They account for about 75 to 85% of the circulating antibodies. They're the main antibody for both secondary and late primary responses. And then the E is actually pretty much like for instance, the stem, okay, the stem of this antibody here binds to mast cells and basophils. And the antigen binding sites, okay, will be the one that triggers, when they get into the receptor, triggers these cells to release histamine and other chemicals that would mediate inflammation and allergic reactions. So the E is secreted mostly by plasma cells in the skin, the mucosa of the gastrointestinal tract, okay, and the level of immunoglobulin E would rise in severe allergic attack or chronic parasitic infection of the gastrointestinal tract. So, plasma, meaning the cloned B cells, can switch from making one class of antibody to another, depending on what they are capable of doing. If they carry the genes to do it, they can switch. They retain specificity for same antigen, and the immunoglobulin M is released during the primary response, but the plasma cell can switch to IgG for secondary response. So, what are the functions of your antibody? Well, the defense mechanism is using antibodies for neutralization, agglutination, precipitation, and complement fixation. Neutralization usually is the simplest and one of the most effective and defensive mechanism. Here, the antibodies block specific sites on viruses or bacterial antitoxins. They prevent antigen from binding to receptors on tissue cells. So the antigen-antibody complex would undergo phagocytosis. Agglutination is actually when antibodies bind to their determinants on two different antigenic markers at the same time. And then they become heavy, okay, and they form complexes to form large lattice-like clumps. And this process is what's called, the clumping together is what's called agglutination. When they agglutinate, then it becomes very easy for the phagocytes to eat those foreign foreign antigens. Precipitation is happening with molecules instead of cells. Okay, soluble molecules are cross-linked into the complex of antibodies. The complex would be precipitating out of solution, and the precipitated complex are now easier for phagocytes to engulf. Remember that this precipitation is actually acting and is actually happening only when antibodies react to soluble molecules, not cells. Then complement fixation and activation. This is actually the main antibody defense against cellular antigens, like whenever there's a mismatch in what it calls blood transfusion or bacteria. Whenever several antibodies are bound close together on the same antigen, complement binding sites on their stem regions are actually aligned. Okay, and this happens most especially during amplification of inflammatory response, for instance, in opsonization. And here, guys, is a good picture of what the functions or the mechanisms of antibody action is. You could pretty much like study it, please.